So I got to know Ashley first when she was a master's student at the University of Delaware. She worked in Charles Bartlett's lab doing a taxonomic revision of a group of plant hoppers. She then changed course for her PhD and worked in Doug Tallamy's lab investigating birds' insect prey preferences. And many of you may have seen her great talk on what do birds eat. Very interesting work. But all along, as I understand it, her first love really was medical entomology. And in fact, at UD in the fall of 2018, she taught a course on medical veterinary and forensic entomology, which was really well received. And when she completed her PhD, she got a postdoc at the tick-borne disease laboratory of the Army Public Health Center in Edgewood, Maryland. Very recently, she's accepted a job as a tick biologist with DENREC. So with that, we're happy to hear Ashley talk about ticks.
Thank you so much for that introduction, Dr. Huff-Goldstein. So I'll just dive right in because I have a lot to say, and I can really ramble on. So if my time's running out, just don't be afraid to jump in and cut me off. So I was going to talk about the ticks of Delaware. I'm going to give an introduction to some general tick ecology, then an overview of some of the species that are most common here, some of the pathogens and other health problems associated with them. And then I'll talk about the Delaware tick program that just started last year, what we're doing about ticks in Delaware, and then I'll wrap up by saying what you can do about ticks in Delaware, and hopefully to limit your encounters with them. So I'm going to give a shout out here to Professor Donald McCreary, who was with the University of Delaware for 38 years, actually before my time, but he's the original Delaware tick expert, and he wrote the first and to date the only report on ticks of Delaware, which I'm going to share some of the information from that report. This was published in 1945. I figured since it's been 3/4 of a century, it's time for an update. So at that time, 75 years ago, there were only 8 known tick species in Delaware. And today, just to compare real quick, we know of 13 species. Now, some of these probably were around back in 1945 but hadn't been detected yet, but a couple of these were ones that invaded from other regions. And back in 1945, we were only aware of one tick-borne pathogen in this area, and that was Rickettsia rickettsii, the bacteria that cause Rocky Mountain spotted fever. The first Delaware case was reported in 1933, but by the time of McCreary's publication, there had been about 50 cases, and it had an 18% fatality rate at that time. And today, we still do have Rocky Mountain spotted fever reported in Delaware occasionally, but what's really incredible is the number of additional pathogens that have been detected since then. So we now have agents that cause anaplasmosis, babesiosis, Lyme disease, tick-borne relapsing fever, Ehrlichiosis, and then 2 that I included, which are not yet reported from Delaware, but I suspect that they do exist in our ticks because they've been reported from our neighboring states, Powassan Virus and Tidewater spotted fever. And this timeline just kind of shows not just the number of pathogens that we're aware of, but how the rate of discovery of new pathogens has really increased in recent decades. And this is kind of interesting. There was a study done a few years ago, a survey by Delaware State University and the Delaware Division of Public Health, and they found that only 38% of respondents felt that ticks are a problem in Delaware, and only 13% strongly agreed that Lyme disease is a problem in Delaware. And as I'm sure most of you know, it's a pretty serious problem. All right, so I'm going to talk a little bit about generalized tick ecology. So what I'm going to say is true for most of the species of medical importance in Delaware. So most of them are 3-host ticks, and that means if they're going to complete their life cycle, they must find a host and take a blood meal from a host 3 times. So they start out as eggs, the eggs hatch into larvae. The larvae quest for a host. Quest is a technical term. It refers to when they crawl up on vegetation and stick out their first pair of legs to grab onto a host walking by. And they will feed for several days, up to a week, and then they drop off and molt into the next stage, which is called the nymph. The nymph is still immature. And they also quest find a host, take a blood meal, drop off the host and molt, and now they're finally adults, and the adults will quest one more time. The females will take a blood meal and the males will mate with them while they're on that third host, and then they drop off and the females can lay eggs with all the protein and resources they got from that last blood meal. And the reason that ticks are such good vectors, or in other words, why they're such bad news for people, is as I just mentioned, they have to take multiple blood meals to complete their life cycle. So that means every time they're taking a blood meal, that's an exposure where they might pick up pathogens. And then if we wind up being one of their subsequent hosts, they can pass those pathogens on to us. They're generalists, so they'll feed on just about any vertebrate species. And that makes it possible for a pathogen to jump from one species to another. Sorry, my dog's saying hello. And that's called a zoonosis, when a disease or when a pathogen jumps from one species to humans. They feed for a long time, at least a few hours, but more likely it's going to be days. And that increases the chances of transmission. The longer they're attached, the more opportunity there is for the pathogen to be transmitted to us. And they can get around easily, not on their own, but by hitchhiking on wildlife, especially birds. So migratory birds bring at least 50 million black-legged ticks into Canada each year. That's just an example of how mobile they are. And then lastly, of course, they're very tough. So if you've ever tried to kill a tick, you know you can't squish them easily. They're protected by a shield. And then they can survive tough conditions. They're not very sensitive to heat or cold. They can survive through cold winters and hot summers. The one thing they're a little bit sensitive to is desiccation, so they don't want to dry out. And there have been a number of factors that are responsible for people encountering ticks more in recent decades than they used to in our earlier history. But one of those that I wanted to address is climate change, which is allowing ticks to spread into new regions by making those places more hospitable for them. So around the world, as the planet warms up, more and more places are becoming suitable tick habitat. And you would think maybe that's being balanced out by some places becoming too hot for ticks, but unfortunately, that's not the case. We're seeing just a net increase in places that are tick habitat because they are very tolerant of hot conditions. So we're seeing them climb northward, and then they're also climbing up in altitude as well. So this is the most common tick in Delaware, and especially if you're in Kent or Sussex County, this would be the most commonly encountered tick for you. And I will say, as the Delaware State tick biologist, I've only been in this role for less than 2 months, but every call or email I've gotten from the public has been about this tick. It's a really big nuisance species. And it's a medical concern because it can vector the bacteria that cause Ehrlichiosis. And it's also associated with alpha-gal syndrome, which is also called the red meat or mammalian meat allergy. And even if they don't infect you with a pathogen or induce an allergy, their bites are just really nasty. They tend to get really itchy. And for each species, I'll show you how to identify them. So this species is easiest to identify if you get an adult female because they have a white dot on their scutum or their shield on the back. But even if you get a male, you can recognize it by the general body shape. It's the roundest tick that we have in Delaware, whereas some of the others are more oval or teardrop shaped. And the mouth parts are an important feature. Amblyomma ticks have the longest mouth parts of any ticks, at least in Delaware. And the males have little streaks of white on the margin or the edge of their shield. And in males, the shield covers their entire back, whereas on females, you can see it just covers sort of the top part of the first half of the back, and that allows them to expand the sac-like part of their body when they're engorging with blood so they can lay eggs. All right, so the American dog tick is probably more familiar to people in Newcastle County. It's a little bit more common in the northern part of the state. And as McCreary had noted back in 1945, it's associated with Rocky Mountain spotted fever and also tick paralysis, which is a condition where an allergic reaction to tick saliva can actually cause your muscles to become paralyzed, and it can be fatal if the tick is not found and removed in time. It can even paralyze the muscles that you need to breathe. And as the name dog tick would suggest, they do prefer dogs, but they will readily bite people. And again, McCreary considered this the most dangerous tick species in Delaware, but it's now pretty low on our priority list, not because it's become less dangerous per se, but just we have other problems to worry about now, other ticks. So the dog tick can be recognized if you look at the shield or scutum. It tends to be pale and marbled, so it's lighter than the rest of the body, and they have very short mouth parts and a relatively large body size. And part of that is that whenever people encounter a dog tick, it's usually an adult tick. People very rarely get the larvae or nymphs on them. And that's in contrast to the other species we'll talk about, which are happy to bite humans at any life cycle stage. The black-legged tick, probably the most notorious tick in our region because of its association with Lyme disease. It can also transmit other pathogens that cause Babesiosis, Anaplasmosis, and Powassan Virus. And most people who get sick from the bite of a black-legged tick are infected by a nymph. And the nymph is actually less likely to be infected than an adult because it's taken fewer blood meals, so it's had less exposure to pathogens. But the nymph is so small and hard to detect that it can often stay attached long enough to transmit pathogens. So you can recognize it by its teardrop shape, its general small body size, and then the shield is darker than the rest of the body. And just to give an idea of how small they are, there are 5 black-legged ticks on this muffin, and it's very difficult to find them all. And I'm going to just take an aside real quick to talk about where the name deer tick comes from, because you'll notice I keep saying black-legged tick, and that's the same species that you've probably heard of as a deer tick. And what happened is back in 1821, a scientist named Thomas Say described the black-legged tick for the first time, and he gave it the name Ixodes scapularis. And then 150 years later, some other scientists described what they thought was a new tick species, and they called it Ixodes damani, the deer tick. And just a few years later, 14 years later, it was determined that they were actually the same species. And of course, whenever that happens, and it happens all the time in taxonomy, the older name is the one that takes precedence. So the one that is most scientifically correct to use is Ixodes scapularis, the black-legged tick. But of course, in those 14 years between 79 and 93, there was an explosion of interest in this tick and in Lyme disease. So a lot of seminal papers on Lyme disease refer to Ixodes damani, the deer tick. And so that name is still used in a lot of public health messaging. So if you talk about deer ticks, everyone will know what you're talking about, but I am going to stick to using black-legged tick just because I prefer using the correct taxonomy. All right, and the last tick I'm going to talk about is the Asian longhorn tick, Haemaphysalis longicornis, which is native to East Asia, but it has spread to Australia, New Zealand, some of the Pacific Islands, and the United States, probably through movement of livestock. It was first detected in New Jersey back in 2017, and it was found in Delaware just 2 years later. Some good news is that this is not a major human biter, but in its native range, it is associated with many human pathogens. So far in the United States, it's only been associated with one pathogen, which is called Theileria orientalis, which is the agent of Borreliosis in cattle. So this is pretty bad news for our cattle industry. So at this point, it is considered mostly a veterinary concern, but we're worried that it might become a medical concern as well, because at least in a laboratory setting, it was shown that it could vector the bacteria that cause Rocky Mountain spotted fever. Next up, I'm going to start talking about the different pathogens and diseases associated with ticks. For each one, I've got a picture in the upper left that shows you the species that transmits it. So this one, the vector is Ixodes scapularis. And the causative agent, or the pathogen, is Borrelia burgdorferi. The signs and symptoms can be incredibly variable between patients. If you catch Lyme disease in the early stage, you'll probably notice fever, aches, fatigue, and the famous erythema migrans, or the bullseye rash. The rash is the only symptom that is so specific to Lyme disease that it's considered diagnostic of Lyme disease. But I should absolutely point out that not all patients get the rash. In fact, not even a majority of patients get the rash, and it can present very differently in different people. And if you get the rash, that's actually a blessing in a way, because that tells you that you have Lyme disease and that you're still in a window where treatment will be most effective. So the more serious problems from late stage Lyme disease happen when that the early stage is not treated appropriately or not detected. And late stage Lyme disease can be marked by just a whole spectrum of neurological symptoms, cardiovascular symptoms, musculoskeletal involvement. So the most commonly reported symptom of late stage Lyme disease is arthritis. In the early stage, it is considered highly treatable with antibiotics. In the late stage, there's not a good medical consensus on what the best treatment would be. And just as an interesting side note, we think of this as being a pretty recent disease because it really only got in the public health spotlight in the 1970s and 80s, but it is actually an ancient disease that's been with us for all of human history. In fact, the famous Tyrolean Iceman from more than 5,000 years ago had Borrelia burgdorferi bacteria. 82% of tick-borne disease cases in the United States between 2004 and 2016 were Lyme disease. So this really is, by the numbers, the most serious one we have to worry about. The CDC estimates 330,000 new cases in the US each year. And Delaware is pretty consistently among the top 10 states for Lyme disease by incidence. And our annual incidence rate is about 67 cases per 100,000 people. And I can't talk about Lyme disease without talking about the ecology a little bit, because there are a lot of environmental or ecological factors that govern the transmission of Lyme disease. So we know that black-legged ticks are generalist feeders. They feed on more than 100 vertebrate species, but not all of those hosts are equally likely to infect the ticks. So a competent vector is a species that is highly likely to infect a tick if the host is infected. And in our area, the most competent reservoir species are white-footed mice, chipmunks, and shrews. So if they happen to have Borrelia burgdorferi bacteria circulating in their bloodstream and a tick feeds on them, the tick will probably pick up the bacteria and in turn be infected and be able to pass on that infection to other hosts. And at the other end of the spectrum, there are lizards, which are incompetent vectors. They actually have a factor in their blood that kills Borrelia burgdorferi, and a tick that feeds on a lizard will be cleared of infection. So this helps to explain the geographic distribution of Lyme disease. As you'll notice, much more common in the Northeast and in the Midwest, where ticks are more likely to encounter a rodent and feed on a rodent as opposed to feeding on a lizard. Whereas on the West Coast and in the Southeastern United States, where they do have ticks that can transmit Lyme disease, those ticks are more likely to encounter a lizard and be cleared of infection and not present a threat to humans anymore. Another important species in the ecology of Ixodes scapularis is the white-tailed deer, which are the reproductive host. So I mentioned in their 3-host life cycle, the 3rd host is the one that they mate on. It's considered the reproductive host. So I just bring that up because many control methods focus on deer and mice. And the last thing about Lyme disease I want to draw attention to is racial disparities in diagnosis and treatment. So if you do a search for Lyme disease, you'll probably see picture after picture of this perfect bullseye rash, usually shown on pale skin. And unfortunately, I already mentioned that most patients don't get that rash, but another factor to consider is that the rash presents very differently on different skin types. So this is an example of what it might look like on someone with darker skin. And the unfortunate result of that is that Lyme disease in its early stage is less likely to be diagnosed in people of color. And so there's a higher rate of late stage Lyme disease marked by arthritis in those patients. So of course, ticks don't discriminate in who they bite, and the bacteria don't discriminate in who they infect. So the discrimination comes in where in our public health messaging, we're not doing a good enough job showing that Lyme disease presents very differently in different people. All right, so switching gears, here's another disease that's associated with black-legged ticks. It's called Anaplasmosis. The causative agent is Anaplasma phagocytophilum. This is another bacterial agent. It's marked by fever, headache, muscle aches, some other symptoms, and it is considered treatable with doxycycline or other antibiotics. Moving right along, another disease associated with black-legged ticks is Babesiosis, caused by Babesia microti. And what's really different about this one is that it's not a bacterial illness. It's caused by a protozoan parasite. So it's more closely related to the agent that causes malaria. And it's marked by malaise and fever, possibly enlarged spleen or liver, some other symptoms. And an important note about this one is that it can also be spread through blood transfusion. So if you've ever been diagnosed with babesiosis, you are no longer eligible to donate blood. And an important point is that because it's not bacterial, it cannot be treated with antibiotics. And this can be a problem because kind of the go-to treatment for someone who's bitten by a tick is to prescribe antibiotics. But in fact, you would need antimalarials to cure babesiosis. So the typical treatment is a combination of atovaquone and azithromycin. And the last disease I'm going to talk about that's associated with the black-legged tick is Powassan virus. And an important note about this one is that it can be transmitted in as little as 15 minutes of attachment. So you've probably heard that ticks need to be attached to you for 24 hours or 48 hours or even 72 hours to transmit a pathogen. And that's it's true that the longer they are attached, the more likely they are to infect you. But Powassan virus can be transmitted in a much shorter window. And because it's a virus, we don't have any specific treatment for it. So you can treat the symptoms, try to make the patient feel better, but you cannot do anything to kill the virus. You just have to wait and see if the patient gets better. And it can have some pretty serious neurological symptoms, encephalitis, seizures, aphasia, or difficulty understanding speech. And as you can see from this map, there have been no neuroinvasive Powassan cases reported in Delaware, but it is in some of our neighboring states. This next one, as you can see, the picture in the top left has changed. So this disease is one that's associated with the Lone Star tick. So Ehrlichiosis is caused by several different species, all in the genus Ehrlichia. And like we've seen in a lot of our other tick-borne illnesses, the symptoms are kind of generalized flu-like symptoms. So if you get flu-like symptoms in the middle of summer, you should consider a tick-borne illness. And this one is bacterial, so it can be treated with doxycycline or other antibiotics. Next up is Rocky-Mounted Spotted Fever, and this one is vectored by the American Dog Tic and then possibly by the Asian Longhorn Tick. We're not sure yet. And it is marked by fever, headache, rash. I included a picture of it earlier that showed that really distinctive rash, nausea, vomiting, muscle pain, and stomach pain. It is treatable with antibiotics, and although cases have been increasing in recent years, fatality rates have declined, and that's probably due both to improvements in diagnosis and treatment, but also the tests that are typically used are very sensitive without being very specific to Rickettsia rickettsii, so they can pick up other rickettsial species that are less pathogenic. So someone might be sick because they've been infected with a different rickettsial species that's not as serious as Rickettsia rickettsii. Next up, and this is the one I think people are most interested in right now because it's pretty new, alpha-gal syndrome is not caused by a pathogen, but it is something that starts out with a tick bite. So alpha-gal is a carbohydrate. It's a sugar that's found in all mammals except for some primates. So if a Lone Star tick feeds on a mammal and then subsequently feeds on a person, they might have some lingering alpha-gal in their alimentary tract, and then they will inject it into the human bloodstream while it's feeding. And when it's injected into the bloodstream, the human immune system recognizes it as a foreign invader and releases antibodies to attack it, and that presents as an allergic immune response. And even if that person has been exposed to that carbohydrate many, many times in their life by eating red meat, when the alpha-gal enters our body through the usual route, through the digestive tract, the body doesn't recognize it as a foreign invader, but then after having been bitten by the tick, our bodies become more sensitized to it and will react to it even when it's just coming in through our diet as usual. So future intake of mammal meat and sometimes dairy products can trigger the allergic reaction. And it's different from most food allergies because the reactions are delayed for hours after ingestion. And it presents with a whole range of allergic symptoms, and there is no specific treatment for it. The best clinical guidance at this time is just to avoid mammalian meat. And then if symptoms persist, also avoid dairy products. And then you might have to do some sleuthing to see if any of the other food or pharmaceutical products in your day-to-day have gelatin or other mammalian byproducts. And they can turn up in some unexpected places, even toothpaste or the cancer-fighting drug cetuximab may trigger an allergic response. And I've talked to a lot of people who have this illness and are very upset about it. I will just say, just as a silver lining, both the quality and the availability of meat alternative products has really skyrocketed in recent years. So I'm not trying to undermine the seriousness of these symptoms, but I'll just say it is easier to avoid red meat today than it was 5 years ago or 10 years ago. So I'm going to talk briefly about the history of our tick program. So it started with the establishment of the Lyme Disease Prevention Task Force back in 2014. And that task force submitted their final report to the General Assembly and the governor in 2015. And that led to the General Assembly authorizing a tick program in 2016. And last year, Dr. Lauren Maestas served as the first state tick biologist for Delaware. And he did just a really fantastic job getting the program off the ground. And I was really excited to step into that role earlier this year. And the main thing that I've been doing right now during these first couple of months is active surveillance. So that means I'm going out looking for ticks. So I'm conducting tick dragging in all 3 counties and 12 different state parks, and then 2 wildlife refuges as well. So tick dragging basically means you're dragging a 1 meter square piece of white cloth over the vegetation, and if there's any ticks questing in that vegetation, they will latch onto it. And then it's easy to just remove them, put them in ethanol vials, and then I take them back to my lab to identify under a microscope. Later in the year, I'll be doing some mammal trapping in the state parks, and then also collaborating with some of our other fish and wildlife professionals to collect ticks from hunter check stations. So here's just a couple of photos from on the job. So those are the contents of a typical tick vial that I got during a drag at a state park, and that's what they look like when they're under the microscope. I'm also doing passive surveillance, which means that as opposed to me going out and getting the ticks, they come to me through participating veterinary clinics and wildlife rehab centers, as well as some farms and stables. And then I've also just asked my colleagues and associates through Fish and Wildlife, and also through University of Delaware and neighbors and just other associates to give me their ticks that they encounter on them or on their pets. And then the ornithologist at Delaware Nature Society, Ian Stewart, has been collecting ticks for me off of birds at his bird banding station. So I'm just going to zoom in on this picture real quick because it's really interesting to see. There's at least 4 ticks on this eastern towhee, and they in turn have little mites on them. So that's an example of hyperparasitism, so a parasite on a parasite. And then the next step after collecting the ticks is to test them to see what pathogens they might be carrying. So that's all going to take place at Deltec Community College. I'm really excited by the partnership we have with the lab of Dr. Michael Buoni, who is an expert molecular biologist. So he is going to screen all of those ticks collected from tick dragging for a variety of pathogens that we might have in this area. So that's our first important collaboration. And then I'm just going to mention some of the other people who are helping me out, because technically the tick program is only one person, it's me, but I don't feel too weighed down by the responsibility because I have so many people helping me out from other institutions. So Dr. Michael Buoni, and then Dr. Vincenzo Ellis at the University of Delaware is starting a black-legged tick ecology project. I'm also working with Dr. Shahid Karim at the University of Southern Mississippi on an alpha-gal syndrome project. So basically, I sent him live Lone Star ticks that I collect in Delaware, and then he's comparing them to the ticks in Mississippi to see what's different between them and if the ones in Delaware are more or less likely to give people alpha-gal syndrome. And then lastly, Dr. Maria Duke-Wasser at Columbia University and her colleagues at Wisconsin-Madison started this great initiative called the Tick app. And I've recently become a partner on that project, so I'm trying to promote it. So a little bit about the Tick app. It's a free smartphone app that you can get anywhere that you get your apps. And it can give you great general information about ticks, frequently asked questions, how to prevent them. And it will ask you to complete a daily log on your outdoor activities. And that helps them to crowdsource data from people all over the country to see, to learn more about patterns of tick exposure. And then what you get in exchange for submitting that log is you can send in pictures of any ticks that you encounter, and they will be identified by experts. So tick identification is really important because, as we saw earlier, each species is identified with, or is associated with, different risks to human health. All right, so I'm going to wrap up with just talking about what you can do to limit your tick exposure. And the first thing I'll talk about is what you can do around your yard. And I'll start out, I know you are gardeners, so you probably don't want to cover your property in concrete, which would be the only way to make your yard 100% completely tick safe. And I don't want anyone to do that. I'll be clear about that. So I started out with this quote from the Connecticut Agricultural Experiment Station Tick Management Handbook, which says, "Elimination of woodland and wildlife habitats is not necessary or environmentally desirable." And in fact, if your yard is a good wildlife habitat, that could potentially help, because having high biodiversity or high species richness, through something called the dilution effect, it functions to keep the relative abundance of white-footed mice low. So white-footed mice are more likely to give ticks the Borrelia infection compared to other species. So if your yard had very, very low diversity, say it only had like 10 species in it and one of them was the white-footed mouse, then a tick that's questing for a host is very likely to encounter a white-footed mouse and feed on it. But if you have a lot of other species in the system, the tick is more likely to encounter something else that is less likely to infect it. And then, of course, as you're adding more species to your yard, you can also add species that will prey on white-footed mice or compete with them, or in other ways help to keep their numbers low. And so, I don't want you to get rid of your wildlife habitat. What you can do is identify the areas of your yard that are used the most frequently. So think about like where kids are playing or where you're going to have your friends over for a cookout, whenever we can have people over again. And you can make those specific zones less suitable for ticks by having lawn there. And it doesn't really matter if it's short grass, or it doesn't matter how short it is, as long as it's lawn and it's in direct sunlight, it will not be suitable tick habitat. There was a study done by Vince D'Amico at the University of Delaware that found that whether you mow your lawn every week, every 2 weeks, or every 3 weeks, it doesn't matter. As long as it's lawn, it's not really good tick habitat. And in those areas, you can also remove leaf litter and brush piles that would make good habitat for rodents. You can reduce the ground cover and make your trails wider and trim branches to let in more sunlight. So here's just a quick example of, they took this swing set, took it away from a woody edge, which would be great tick habitat, and put it in direct sunlight, and then also remove the vegetation underneath it. You can also help by removing invasive plant species like multiflora rose and Japanese barberry. And there have been numerous studies that show that these species in particular help ticks out because they provide a humid and a stable microclimate that is conducive to tick survival. So here's just a picture from one of those studies. So sites that had barberry that was not controlled had a much higher density of infected black-legged ticks compared to sites where the barberry had been killed, and then sites that never had any barberry to begin with. And then this next one I know is not very practical for most people because it's so difficult to achieve, but if you can exclude deer from your property through fencing or through other measures, we talked about how deer are the reproductive hosts for ticks, so their populations are linked. And if you can't put up fences, you can try deer repellents or planting deer-resistant plants. And I know that you probably know as gardeners that sometimes the deer don't know that they're supposed to avoid the deer-resistant plants, but just mentioning it is something you can possibly try. Next, I'm going to dispel a myth really quick. A lot of people are hopeful that if they have chickens or guinea fowl on their property, that these will help to keep tick numbers low. And unfortunately, that's not really substantiated. It's true that these birds and then other wildlife species like opossums will eat ticks opportunistically. So as the opportunity presents itself, for example, while they're grooming, they might eat any ticks they encounter, but it's not like they're combing through your property trying to eat all the ticks they can find. So they're not a specialized enough tick predator to actually achieve tick control. So the next thing you can do is just to think about personal protection. And I know you've probably heard these tips before. They're pretty well communicated, I think. But there's a survey that found that even though everybody knows these tips, they don't often practice them. So if we could just raise compliance and just get into a routine of making sure that you actually follow these rules when you go out, we should see numbers of tick-borne infections drop. So you can wear repellent, and if you're putting it on your skin, the most effective active ingredient is DEET, and if you're putting it on your clothing or shoes, the CDC recommends permethrin. I will point out that permethrin is sensitive, or it's dangerous for cats, so try to limit their exposure to your treated clothes or shoes. And then in my day-to-day, the thing that I noticed that made the biggest difference in the terms of how many ticks I got was when I started wearing tall rubber boots instead of wearing sneakers out in the field. It just had a really dramatic effect because most of the ticks that are questing, they're questing at that boot height. So it's just giving them a surface that's difficult for them to grab onto compared to clothing. And you can wear long sleeves, tuck your pants into your socks. If you wear light colored clothing, it makes it easier to spot the ticks and remove them. This is a picture of my leg from last week out in the field. All of those are ticks. You can keep your pets safe by checking them for ticks regularly and keeping them up to date on tick preventatives. And then make sure that you're checking yourself for ticks regularly. So after you've been in tick habitat, whether that's your yard or a park or whatever, come indoors and take a shower. And I want to point out that the shower itself is not going to remove the ticks. Running water isn't enough to dislodge them if they've become attached to your skin. But the point of taking the shower is to give yourself an opportunity to really examine all your nooks and crannies. So if you find ticks, you want to remove them as quickly as possible. I've met people who will make an appointment with their doctor to remove a tick for them, and I think that's just ridiculous because the doctor's not going to do anything that you can't do yourself. And the longer the tick is attached, the higher the likelihood of pathogen transmission. So if you do get a tick, you can just use fine-tipped tweezers to grasp it as close to the skin surface as possible. I often don't have tweezers on me when I find a tick, and I just use my thumb and finger. And I pull upward with steady, even pressure. Just pull it away from the skin, and you try not to twist it or do it too quickly because you don't want the mouth parts to break off in the skin. But if that happens, it is not the end of the world. It doesn't mean that you would be at an increased risk of a tick-borne infection. It's just akin to having a splinter in your skin. So it makes it a little bit harder for that bite wound to heal, which could leave you vulnerable to secondary infections. But again, it's not going to mean that you're more likely to get Lyme disease or another tick-borne illness. And then wash the bite site and wash your hands. I would save the tick specimen for reference. See if you can get it identified either yourself or by an expert. There are also facilities that conduct pathogen testing if you want to have the tick tested. And as far as next steps, the most cautious approach. So if you wanted to be absolutely safe, you could talk to a medical professional after a tick bite, and they would probably recommend a course of prophylactic antibiotics. So even in the absence of symptoms, they would say, take this course of antibiotics. And I know that that approach isn't practical for everyone. For me, for example, I get ticks all the time and it's just not practical for me to be on antibiotics all the time. So when I get a tick, I just remain vigilant for symptoms of tick-borne illness and consult a doctor if I get any flu-like symptoms. And I should point out at this juncture, I am absolutely not that kind of doctor. So, you know, please talk to your doctor if you have any concerns. And then if they do prescribe antibiotics, make sure you finish the entire course. And lastly, I'll just leave you with some resources. Again, the Tick app is a great citizen science tool that can also answer your questions about ticks and get your ticks identified for you. I mentioned a lot of tips that I got from the Tick Management Handbook, which anybody can download. The Tick Encounter website has some great images, and what I really like about it is it also includes images of ticks that are engorged, so they look totally different when they've been engorged with blood, and it's the only site I found that has pictures of that for every species that is commonly encountered here. And then the Northeast Regional Center for Excellence in Vector-Borne Diseases has a lot of more in-depth information available. And then lastly, I'll leave you with my email address in case you have any questions for me or questions specifically about Delaware ticks. So thank you very, very much for your time and attention. I'm sorry I rambled on for so long.